Hello, everyone. Greeting from ARSHI. I hope everybody's doing well. Thank you for joining the webinar today. And it's our honor, ARSHI is honored today to have Dr. Faisal Khan is our guest speaker, and our moderator is Dr. Muhammad Lawaji. Without further notice, I would like to welcome you and give the floor to Dr. Muhammad to introduce our speaker and to start the talk.
Thank you very much, Fadi. Good evening, good afternoon, and good morning, everyone. It is my pleasure to introduce our speaker today, Dr. Faisal Masood Khan. And before I start my introduction, I want to ask everyone to kindly mute yourself during the presentation. We will have around 15 minutes toward the end allocated for question and answers. You can write your question and comment in the chat section, or you can raise your hand if you wish to speak. Our speaker for today is Dr. Faisal Khan. I had the pleasure of working with and learning from Dr. Khan during his consultation for our laboratory in Dammam between 2009 and 2014. Dr. Khan obtained his doctoral degree from India and completed his postdoctoral training and clinical fellowship in Calgary, Canada. With close to 20 years of experience in transplant immunology, cancer genomics, and histocompatibility, Dr. Khan currently continue to serve as an associate professor in Cumming School of Medicine at the University of Calgary. He also holds the Barb Ibbotson Chair of Pediatric Hematology, as well as the Associate Clinical Director for the Histocompatibility and Immunogenetics Laboratory, and Scientific Lead of Molecular Pathology Program in Alberta Precision Labs. Dr. Khan's research effort has been recognized in form of awards in several occasions. His research work has yielded 87 research articles published in journals such as Science, Blood, and many other reputed scientific journals. With no further ado, I give the mic to Dr. Khan.
Thank you very much, Mohammed, and thank you very much, Arshi, for asking me to present in this forum. I was one of the founder members of Arshi. I still remember the time when we started this first time during a discussion in Chicago, and it's lovely to see how far we have came from that time. So focusing specifically for today's talk, I have titled it as a mathematical equation of 14 plus 5 is equals to 0, an immunogenetic algorithm for precision medicine. A little awkward topic, and I hope that I will be able to justify this topic during the course of the presentation today, and try to take the broader prospects of the work done in the histocompatibility and immunogenetics arena as not being just focused for transplantation, but much beyond than transplantation. So it's trying to my apologies. I'm trying to work on the slides.
The bottom line, the bottom left. You see the arrows?
Yeah. Okay. The word HLA or its role in transplantation or in broader histocompatibility field has been there for ages. But somehow an event in the year 2001, when the first draft of human genome sequence became available, has really energized with what we could do using different tools for genomics. And like every other field in genomics, the field of histocompatibility and immunogenetics also moved significantly forward since that time. And among different findings that became available after the sequencing program, the most important one was genetic diversity. which shows that if we randomly select any two individuals, then genetically they are 99.9% similar. Obviously, a little heartbreaking for the geneticists all over the world that we just have to deal with 0.1% of genetic difference existing between two individuals. But if we look a little deeper, then that 0.1% difference roughly corresponds to 3 million nucleotide differences between two different individuals. And what these differences can do, they just not give us different phenotypes, but they could lead to different susceptibility to diseases, different immunity to infections, different actions of drugs, and different outcome of a treatment. For the sake of today's presentation, different outcome of transplantation. And that's pretty much the basis which I want to cover in today's presentation. There's a little bit of bias where I will be mainly showing the data that came out from our group, but I have made every attempt to compare and provide a comprehensive picture using some of the information and data published by several groups across the world in the next few slides. So coming back to the title. So as I said that this is basically an immunogenetic algorithm for precision medicine. And what does that mean is, I'll start with the simpler one. The 5 corresponds to the classical HLA class 1 and 2 molecules, HLA A, B, C, DR, and DQ. The 14 corresponds to killer immunoglobulin-like receptor gene complex. And the 0 is failure of a treatment. So the whole idea of the work that I'm going to cover today revolves around how the combination of KIRs and HLA can provide a precise model of medicine where the chances of the failure of a treatment is nearly 0. And I'll start first a little bit about what these KIR HLA molecules are responsible for. So as we all know, it's the natural killer cells. Natural killer cells, they correspond to around 10 to 15% of all lymphocytes. They neither have memory nor they need prior sensitization to start an immune response. They are the first line of defense against tumors. They have very strong antiviral activity. And once activated, NK cells can perform a variety of target-induced functions like cellular toxicity, ADCC, production of cytokines and chemokines, and many, many more things. And if we look a little more deeper about these natural killer cells, then there are 2 types, the CD56 bright population and the CD56 dim population. And each one of them have their own characteristic features as well as their own functions. So for example, CD56 bright, as the name suggests, has a lot of CD56 molecules, while the CD56 dim population have a lot of CD16 or FC gamma receptor alpha expressed on the surface. The bright population have very low expression of KIRs, while the dim population have very high expression of KIRs. The cytokine receptors are mainly found on the bright population, and the cytolytic granules are mainly found on the dim population. The reason why I wanted to show this subtype differentiation is because these kind of phenotypic differences also lead to different type of functional responses from NK cells. For example, the CD56 dim are often called cytolytic NK cells because their primary function is cytolysis, like CD8 T cells. While the CD56 bright population are often called regulatory NK cells because their primary role is involved in the cytokine production and stimulation. Roughly 90% of NK cells belongs to the cytolytic NK cells type, and that's why the cytolytic function is primarily the major function contributed by natural killer cells. These natural killer cells have several immune checkpoints. I love this term immune checkpoints, which was mainly coined by a group from Dr. Kathleen Su to define the range of receptors that are present on the surface of natural killer cells, and they control how the NK cell operates. And this is that whole view of the different types of receptors present on NK cells. It includes the natural lytic receptors like CD16 and NKP46, NKP30, NKP44. But again, for the sake of today's talk, I'm trying to keep it more focused on what we should be discussing today. This is a group of receptors which are more important because they all interact with HLA class 1 molecules. On the left side, it's the inhibitory receptor, and as the name suggests, they are mainly involved in inhibiting the function of NK cells. And on the right side are the activating receptors, which are mainly involved in activating the natural killer cells. And among these sets of receptor, the most important ones apparently are killer immunoglobulin-like receptors. And as the name suggests, they have immunoglobulin-like domains, the extracellular domains, which gives them the name of killers. They have killing as the primary function. And if you could look at their structure, there are two types, one with a short tail and others with a long tail. The one with the long tail are inhibitory, and the one with the short tail are activating. And if you look at the gene complex on chromosome 19, then typically there are 16 KIR genes, but 2 of them are pseudogenes depicted by the term DP. The letter L represents the inhibition, and the letter S represents the activation, S for short and L for long tails. So in the human population, these KIR genes are known to occur in strong linkage disequilibrium, and they're tightly packed and typically inherited as haplotypes. And there are 2 known haplotypes of KIR, the type A and the type B. The type A haplotype has only 1 activating gene, while type B haplotype can have up to 5 activating genes. And if we look a little more deeper in this gene structure, then you could see that there is a 14 kb repeat enriched sequence that is existing in between the 2 half of the KIR complex, the centromeric half and the telomeric half. And as you could see, the group A, or the haplotype A, has just 1 activating receptor called 2DS4, while group B can have anywhere from 2 to 5 activating receptors. It's much diverse, and it is much activating in function compared to the group A haplotype. And that pretty much suggests the complexity of the KIR gene system. And as I mentioned earlier, that the ligands of these KIR molecules, like many other NK cell receptors, are HLA class 1 molecules. So on the left side is the inhibitory receptors, and on the right side are the activating receptors. And as you could see, the activating receptors can get activated by binding to a variety of ligands. Some of them are HLA class 1 molecules, but then there are several others, and there are so many which are not even known. However, for inhibitory receptors, HLA class 1 molecule are the only ligands that interacts with these inhibitory receptors and inhibits the function of NK cells. Just as a housekeeping thing, when you are reviewing these slides at a later time, I have tried to put a reference on the right corner of my slides, which is something like suggested reading. These are the most recent and most comprehensive reviews that covers the particular topic which I'm presenting on that slide. If you want to have more information about these things. The reason why I picked the most recent one, because they also have the older important reviews and article in their reference list. So please make use of it if you would like to know a little bit more than what I'm presenting here. So, as I said, HLA class 1 molecule corresponds to the primary ligand for the inhibitory KIRs. And in this slide, I will depict how these KIRs regulate and educate natural killer cells. So, for example, this is the NK cell, which is expressing activating KIR, a short-tailed KIR. When this activating KIR finds its ligand on a target cells, it gives the signal for cytolytic granules to come out and lyse the cells. When the similar thing happens, but the NK cell also expresses an inhibitory KIR, and that inhibitory KIR, the long tail one, finds its ligand HLA class 1 molecule on the target cell, then this interaction cancels the signal of this interaction, and it leads to no lysis. And this is how KIR HLA interaction spares the normal healthy cells and lead to killing of tumor or virally infected cells. And this mechanism, known as NK cell education, also known as NK cell licensing, as the NK cell learns who to kill and how to kill during the course of the development, and that education is guided by how they interact with their self HLA class 1 molecules. So using this model, I'm going to show here 4 different prototypes through which KIR-based NK cell regulation happens. The first prototype suggests that if NK cells have a large number of activating receptors present on its surface, then NK cells have a very strong function against the target cells. So the first prototype is based on the high number of activating receptors. The second prototype suggests that there is some specific type of KIRs, here shown in the form of a small crown, that if those KIRs or those activating KIRs are present, then their interaction with the target cell ligands is a lot more stronger, and the activity of NK cells is far superior compared to NK cells who are not expressing that specific type of KIR. The third prototype, as we all know, the one which is very popular, is the missing ligand prototype, which means that it doesn't matter how much activation is there, but if there's a lack of inhibition, when an inhibitory receptor is not able to find its own ligand on the target cell, so there is no inhibition happening here, then the scenario leads to a stronger NK cell function against target cells. And finally, the fourth prototype, which is more linked with the antibody dependent cell cytotoxicity function of natural killer cells, where CD16A molecules interact with an antibody binding against a receptor on a target cells and defining how strong is NK cell mediated ADCC against these target cells. And in today's talk, I am going to cover some of these models in the context of different diseases. So using these 4 prototypes, I would be covering 4 different models of precision medicine. The first one is the transplantation. That how these KIR HLA interaction can be used to improve the immunogenetic algorithm for the selection of donors for allogeneic HCT recipients. The second is cancers, that whether carriage of certain KIRs in HLA could predispose a person to have a protective immune surveillance against hematological malignancies. Third is infections, which means if there's a way that this KIR HLA inheritance leads to predisposition of a strong immune response against the viral infections. And finally, how these KIR HLA interactions or other NK cell related immunogenetics can influence the response of immunotherapy against any type of cancers. So let's start first with the first model, which is how this KIR HLA genetics could be used for improving hematopoietic cell transplant donor selections. And if we look into detail of what is available in literature, then the 3 models have been aggressively shown to be important. The first model, where haplotype B or a higher content of activating receptor will have a strong anti-leukemic effect, has been repeatedly shown from the group of Dr. Sarah Cooley, Dr. Peter Perham, Dr. Miller, Dr. Biesdorf, and I have cited some of the important papers here, but they're just not restricted to these papers. There are many, many more papers that have came out from this team, including some of the clinical trials that either have been completed or ongoing, where the group has in Minnesota has clearly showed that NK cells from donor carrying a haplotype B has a strong anti-leukemic activity and leads to low incidence of relapse after transplantation. They have shown this in the context of acute myeloid leukemia as well as in the context of non-Hodgkin's lymphoma. The second model, where there's a specific here which seems to have importance in an anti-leukemic activity after transplantation. Dr. Catherine Su and Dr. Petersdorf has shown this both in the context of an activating receptor 2DS1 in the presence of HLA-C1, as well as an inhibitory KIR context of KIR3DL1 and HLA-BW4, primarily in the unrelated HLA-matched transplant setting for acute myeloid leukemia. And then the third model, which is the missing ligand model, it was first of all shown in 2002 by the group, the European group from Italy, Dr. Veladi's group, in the context of haploidentical transplant, and has been repeatedly shown in many unrelated transplant settings where a missing KIR HLA ligand scenario between donor and recipient could lead to a strong anti-leukemic activity, leading to low incidence of relapse. And I have put up a review in the Frontiers of Immunology published in 2017, which nicely covers majority of these studies, which have been a lot of replication in different publications, so that they have been consistently shown to be important in terms of donor selection for graft versus leukemia. And using this background, I'm going to show you some of the data that we have generated here in Calgary. There was a paper from a group published in 2012 where we assessed how natural killer cells and other cell subsets recover after transplant. And if you could see here, it's a 90th and the 10th percentile of different lymphocyte subsets in healthy individuals. And as it shows, the natural killer cells are the only lymphocytes that recovers back to the normal level within weeks after transplants. The CD8 T cells take months, and CD4 T cells take years to recover back to the normal level, which itself suggests that the role of NK cells is extremely important. And if we look at the type of transplants being performed in Alberta, we performed around 125 to even 150 transplants per year. 90% of them are 10 out of 10 HLA matched. We use anti-thymocyte globulin as a preconditioning prophylaxis for GVHD for all our patients. And roughly 70% of our transplants are unrelated transplants. And if you look at our outcome scenario, then relapse due to leukemia after transplant is the foremost cause of post-transplant mortality. Roughly 23% of our patients suffer relapse, and almost all of them die. It's almost a death warrant if you have a relapse after transplant. So we took this study and analyzed 281 donor-recipient pairs. Rehan, the research scientist from my lab, has led this study. He's most likely present in today's webinar too. So he studied 281 donor-recipient pairs. Importantly, it was a very homogeneous cohort treated with the same transplant protocol throughout, including the doses of anti-thymocyte globulin. And what we found that in the scenarios when donor inhibitory KIRs don't have the HLA ligands present in recipient, then the cumulative incidence of relapse is far lower compared to scenarios where HLA ligands are present. And such mechanism does not exist for the activating KIRs. So this confirms that lack of inhibition for donor inhibitory KIRs could lead to a stronger graft versus leukemia effect, ultimately reducing the incidence of relapse. Then the second important work, which I'm going to talk a little bit more in detail because it also overlaps with the model of infections here. As I mentioned, all the patients here in Calgary are treated with anti-thymocyte globulin, and anti-thymocyte globulin is a polyclonal IgG against a variety of receptors. And there is a big impact of what kind of prophylaxis is used for the conditioning on how the immunity recovers after transplant. So for example, here in this data, we have shown that when we compare the patients with ATG
you're welcome.
and without non-ATG conditioning, the recovery of NK cells is significantly superior in ATG condition transplants compared to non-ATG condition transplant. And it's more likely due to the ratio of NK cell versus T cell recovery that happens after transplant. So obviously the role of NK cells, if not better, at least different when the transplants are performed with the ATG conditioning. And this is what we saw in the context of infections and the reason of mortality after transplant, that when transplants are done with ATG, incidence of post-transplant lymphoproliferative disease linked with the Epstein-Barr virus-related infections is significantly higher. In other transplant setting, the incidence of PTLD is around 1% or 1.5%, but when they are done in ATG conditioning transplant, as shown from the data from our team, the incidence is around 8.1%. Obviously, PTLD is a very important complication of ATG conditioned transplant, and the mortality rate is anywhere between 30 to 50% in our end. So we took this study where we wanted to assess, is there a role of NK cells through clear HLA regulation that can influence the incidence of PTLD, or in fact influence the incidence of EBV reactivation after transplant. So what we did, we studied around 377 HLA matched transplant donors against again, it's a homogeneous protocol, split it into a discovery and validation cohort, and performed the next generation sequencing of the KIR complex in all these patients. And what was most surprising to us is this result, that when transplant is done with a donor carrying a telomeric AO1 KIR group, the incidence of PTLD is almost zero. And it is significantly higher, almost 25%, when the transplant is done using a donor that does not have a telomeric AO1 KIR group. Because it was a next generation sequencing based protocol, so we were also able to assess the copy numbers. And what we found, that doesn't matter, even if one copy of telomeric AO1 group is present in donor, incidence of PTLD is rock bottom, almost zero. It's 1.8% basically, which was detected. It was less than 1% that was detected in the group with the telomeric AO1 donors. So it was a wonderful finding. But we sat on it. This is still not published. It received an ARSHI Abstract Achievement Award recently, and hoping that it will see the light of the day by the end of this year. The main reason was we wanted to complete this whole story with all the functional mechanism behind this association. So we, so just for the knowledge sake, what the telomeric AO1 group means, the group of these two genes, which are always segregated together, an inhibitory gene 3DL1, and activating gene 2DS4. It is the presence of these two genes that leads to a stronger immunity against PTLD or Epstein-Barr virus. So then we asked the question, is it KIR 3DL1 or 2DS4 or both? And as you could see in this linkage disequilibrium plot, that the 3DL1 and 2DS4 have a linkage disequilibrium of 1, which means it's pretty much they're always together. But then there is a difference, because 3DL1 has a ligand, HLA-BW4. So the question that we asked, that if it is 3DL1 which is responsible, then the effect should be a lot less if HLA-BW4 is present, because that will inhibit the NK cells functioning against the 3DL1 mediated NK cell response against Epstein-Barr virus. So when we look at this, we found no, whether HLA-BW4 negative or positive, donor telomeric AO1 group still have a pretty similar effect on PTLD. Again, suggesting that although it's TOA1, but probably is a 2DS4, which is an activating receptor involved in this work. So we took to the third question, where we wanted to assess what is the functional mechanism. And to assess the functional mechanism, we studied 65 healthy individuals, performed the karyotyping, and assessed the NK cell function against Epstein-Barr virus. Also looked into how the NK cell transcriptome changes when they are challenged with the Epstein-Barr virus transformed cells. So healthy individuals, the karyotyping, the flow-based analysis for the functions, and the transcript analysis. All this work was conducted in the last 2 years in the lab. And what we found, I'll skip the slide, that healthy individuals who don't have telomeric AO1 group have very poor function of NK cells against Epstein-Barr virus, versus individuals who have telomeric AO1 group have a very strong function. And if you compare this with the data that I showed in the transplant setting, it is this group which has high incidence of PTLD, and it is this group which has a very low incidence of PTLD. Again, confirming that if you have a telomeric AO1 group present, the NK cell function is very strong against Epstein-Barr virus. So we took a transcriptome assessment and have a panel that we analyze on nanostring platform, which has NK cell receptors, signaling mediators, and effector molecules. And we assess the function, we assess the transcriptome both before the challenge of EBV as well as after the challenge of EBV. And as you could see, there are some genes which are highly upregulated after the challenge, while there were others which were highly upregulated before the challenge. And when we assessed all these and analyzed it with using different bioinformatic tools, this is what we found. That this is the profile of telomeric AO1 positive individual, and this is the profile of telomeric AO1 negative individuals. When we compare 21 genes get upregulated in all individuals. However, there are 8 genes which get upregulated exclusively if you have this KIR group present. And all of these 8 are involved in the signaling pathway of NK cells. So now, when we're ready to publish this work, this was the last question we asked again. Is it 3DL1 or 2DS4? And we did not want to rely only on the association data that if BW4 is negative, then also there is no impact on the association, suggesting it's likely 2DS4. So what we did, we used the antibodies against specific KIRs. So for example, all KIRs with 1 fluorochrome and 2DS4 with 1 fluorochrome, which makes this group to be positive for 2DS4. Similarly for 3DL1, and when we put the 2 of them together, this is a group which is expressing both the genes of the TAO1 complex, the 3DL1 as well as the 2DS4. And what we found, that the highest function of NK cells against Epstein-Barr virus comes from the NK cells that expresses 2DS4. And it is barely there when only 3DL1 is present on NK cells. So just for the sake of information here, that not all NK cell expresses all the KIRs. Some clones expresses some KIRs and other clones expresses other clones. So this is pretty much the end of what we have accumulated in terms of data for the PTLD story so far. And what it shows, that if donors carry telomeric AO1 group, more specifically KIR2DS4, then it leads to upregulation of signaling molecules. And this leads to stronger NK cell response against EBV. And this is what protects patients from developing PTLD. So if we simply genotype donors for telomeric AO1 group, we may have a protective immune reconstitution in terms of NK cells against PTLD. So overall, for this particular model, this is what we show, that model 2, where there is a specific KIR2DS4 is protective against PTLD, And the prototype 3, the missing ligand, is protective against leukemia relapse. And how it can help in improving HCT donor selection could be specified through this cartoon, that at present, if we have a patient with HLA typing shown here, and we have two donors who are full house HLA match for the 5 loci for this patient, we suggest that immunogenetically, any of these two donors could be used because they are full HLA compatible. However, if you could bring the KIR information in it, then this donor becomes a better donor because this donor has an inhibitory KIR ligand mismatch and also carry a KIR TLOVER1 group, hoping that it will lead to less incidence of relapse and less incidence of PTLD after transplant. So moving on to the second model, which is about the protection from hematological malignancies. Again, there's not much available in the literature because majority of this work has been focused on how relapse of the malignancy could be prevented, either after transplant or through the routine anti-cancer therapies. But whether there is certain type of KIR HLA genes whose inheritance predispose an individual to protection or susceptibility to these cancers is poorly understood, and there's not much in the literature available here. And what I'm showing you here is a data set, quite an extensive data set that Aris from my lab has been working on quite some time. And again, this is an unpublished data. Hopefully we'll be publishing it again this year. Just wanted to finish off a few more patients that can cover up to 2017. So in this study, we have around 837 individuals, including healthy controls and patients of lymphocytic leukemia as well as myelogenous leukemia. And we assess the KIR and HLA1 molecule and analyze all different type of models. And if we look at hazard ratio, the one which are extremely protective are all activating KIRs. And as you could see here, as the number of activating KIR molecules keep on increasing, the level of protection against the hematological malignancy keeps on increasing, suggesting that the model that is more important from the hematological malignancy perspective is the model 1, where higher activating KIR content is conferring protection against the malignancies, irrespective whether it's a myelogenous or lymphoid malignancies, pretty much in tandem with the data shown by Dr. Sarah Cooley's group in Minnesota. And such findings are extremely critical when we are looking at KIR-guided NK cell therapies for hematological malignancies. Coming on quickly, so I have made the last 3 models quite smaller just to make sure that we cover more on transplantation and others, at least some stuff, but not all. So protection from infection and how KIR HLA genetics could play a role here, it's a vast topic because there's so much of data about HCV, HIV, and the CMV viruses available in literature beyond the scope of covering this in this talk. So I have included this, this review information for suggested reading, a wonderful review that covers some of these aspects. What I wanted to show today was more the PTLD story, because there's no such report available in the literature, as well as some of the information about the COVID-19 in this particular model. You know, what we know about NK cells and COVID-19 is summarized beautifully so far. Whatever we know in this review came out in August, which shows that NK cells are more likely a double-edged sword in the setting of COVID-19. On one side, where they should be expressing the CD107A and secreting the cytokines like TNF-alpha, IL-2, and interferon gamma, they should be protecting against the SARS-CoV-2, triggering an NK cell response against the virus. But on the inside, it is shown repeatedly that they express NKG2A, higher expression of inhibitory receptor, which ultimately leads to NK cell exhaustion and releases cytokines like interleukin-6 and 10, which leads to, again, exhaust the T cells and macrophages, ultimately leading to a severe infection of SARS-CoV-2. So which way the story is going, it is still debatable, and there are many, many people across the world who are trying to understand where it goes. And then there's a second part of the same story for how NK cells could be relevant in COVID-19, again, pointing towards that it's a double-edged sword. And if you could see that when the analysis is done for NK cells in the blood vessels, then they are mainly exhausted NK cells, not able to trigger a good immune response. But then if we look at the NK cells in the lung, then they are mainly activated NK cells contributing to inflammation. So again, which way the story is going is still debatable, and I want to share with you some data that would be available most likely next week. So it has been accepted in Blood Advances from our team and should be available for public most likely this week. But here is the data where we have assessed 37 hospitalized COVID-19 patients and 75 healthy individuals. We did an NK cell phenotyping and showed that NK cell counts is severely impaired in COVID-19 patients as compared to the controls. And majority of whatever is left are the cytolytic NK cells. The regulatory NK cells are just not being present in the COVID-19 patients at all. But still, all the right two plot shows the percentages, and the left plot shows the count. So still it shows us 90% of them are cytolytic NK cells, but the count is severely impaired in the severely infected patients. And we looked at their functions, which means NK cell functions against K562 cell lines. And as you could see, again, that the function is severely impaired in COVID-19 patients compared to what has been shown in healthy controls. We also looked at different cytokines, the interleukin 21, interleukin 15, and interleukin 12 in the serum of these patients, and they were just not detectable, suggesting that there is a severe impairment of NK cell functions, counts, and other proteins linked to their functionality in the serum specimen. And that lead us to launch a new study, which we are hoping to complete in next 6 months, much broader study where we are having healthy volunteer, we have mild COVID-19 patients, we have hospitalized severe COVID-19 patients, and as an additional control, we have hospitalized non-COVID infection patients, so patients who have infection due to any other viruses other than COVID-19. We are assessing their DNA profile for KIR and HLA genes. We are doing an NK cell phenotype, and we are assessing their functionality against different NK cell targets, also doing the RNA and cytokine assessment, just to contribute on actually what is the role of NK cells, and if there are specific type of NK cells that confers protection against the severe infection, and whether those specific type of NK cells in the future could be a basis of NK cell-based therapy against COVID-19. Again, very premature, but this is an interesting piece of data that I wanted to share today in this forum. And finally coming, it's not model 3, it's model 4. That is the prediction of immune responses to immunotherapies, which is the last thing that I want to cover in today's talk. What I mean by this, that in different type of immunotherapies, when we have antibodies that specifically target a receptor on a target cell, let's assume the target cell being a malignant cell, then this interaction of antibodies and its antigen or its receptor on the target cell passes through 3 pathways, either ADCC or CDC or apoptosis. And if we look into each of these pathways, then there are molecules which plays a role for which there are known gene variants, which either influences their expression or their function or their interaction with the antibodies. And that's the whole basis of this 4th model, to assess how NK cell mediated ADCC, which includes which includes Fc gamma receptor alpha or CD16A and KIR HLA present on the NK cell and its target cells and contribute to NK cell's response against the target cell. And here's an example. So for example, an NK cell for in the context of rituximab therapy against B cell malignancies. So if this is a B cell expressing CD20 and a patient is treated with rituximab, the constant arm of the rituximab antibody binds with the CD16A receptor on NK cells. And this leads to the release of cytolytic granules and killing of the B cell malignant cells. So this is what we know. But the important point here is this CD16 gene is known to have a variant at a location where valine is mutated to phenylalanine. But if a valine allele is present, then the CD16A has 5 to 8-fold increased affinity to bind with any IgG. In this case, is rituximab. So expectedly, if a patient has a V allele for CD16A, then the rituximab is having a lot more stronger response against the B cell malignancy compared to an individual who has a F allele. So we took this work in the lab where we have a variety of lymphoma cells. Yasin and Aris worked in this on this work, and Yasin actually received a scholar award last year in the ARSHI meeting for this work. That we have a variety of lymphoma cells which we coat with the rituximab and then culture them to assess the NK cell response, both in terms of CD107A and interferon gamma production. And we did this in a series of healthy individuals. And what we found that irrespective of the cell lines, so these are variety of B cell malignancy cell lines, diffuse large B cell lymphoma, Mantel's lymphoma, follicular lymphoma, pretty much whatever we could find available in our repertoire, we use those cell lines as a source of challenge for the mononuclear cells. And universally, individuals who have the V allele or a GG in terms of a G allele in terms of the nucleotides have a very strong NK cell function in response to rituximab compared to individuals who have the phenylalanine allele. And this was universal for all different cell lines. But what was more intriguing was this finding, because all these individuals carried 3DL1. And what we found that if the cell line lacks HLA-BW4, which is an inhibitory molecule, inhibitory ligand for 3DL1, then the functionality is even better compared to function against cell line that do not have HLA-BW4 present in it. And when we put these two models, the CD16 model and the KIR HLA model together, this is what we could show, that the group 1, which has a good CD16 allele and missing KIR ligand for 3DL1, have the strongest response against rituximab compared to a group 4, which has a bad CD16 allele and the inhibition through HLA-BW4 is present in that ligand. So very stark difference between the two groups of patients. And so this is exactly what it is. If you summarize that, you know, the model 4, where CD16 influences an immunotherapy, and model 3, which was so far being mainly reported in the context of protection from leukemia relapse, offers a kind of inhibition where NK cell response could be inhibited if the KIR and ligand both are present, but a missing ligand can have a stronger NK cell mediated ADCC for rituximab. And what's more important here, that whatever I have shown here for rituximab is true in the context of rituximab, but at the same time, there are so many other immunotherapies like trastuzumab for breast cancer, ATG and alemtuzumab for transplant, cetuximab for colon cancer, anti-GD2 for neuroblastoma, or the presence of HLA-DSA leading to antibody-mediated renal graft rejection, all have ADCC mechanism. And the same model can be studied in so many of them. The one with the blue arrows here are the ones which we are studying in our program here, and hopefully at some stage we'll be able to share some more data on it. But there are some literature already started appearing, if you could see, for rituximab, for the CD16, there have been some paper, and Dr. Catherine Sue's group has very nicely shown the role of 3DL1 and BW4, both for denosumab and the anti-GD2 for the neuroblastoma cancers, where the lack of inhibition leads to a lot better response of immunotherapy compared to the scenarios where both KIR and HLA molecules are present. So with that, I would like to summarize whatever I have presented today, that in terms of the first model in transplantation, there are suggestive evidence, quite compelling evidences, that activating KIRs, missing KIR ligand, and specific KIR genes all would lead to improving donor selection criteria for transplantation. Each center with a different protocol need to assess which of these models are applicable to their protocols, because what we have understood is different protocols lead to different pattern of recovery of NK cells. And therefore, which particular type of KIR HLA model is more applicable in that setting needs to be evaluated within that setting, rather than relying on the results produced from a particular group of hospitals. For the cancers, again, there are some evidences appearing, including the one from our group, which shows presence of activating KIRs and presence of some specific activating KIRs predispose an individual to protection from hematological malignancies. For infections, the data I showed you about the 2DS4 or telomeric 1 against Epstein-Barr virus. Similar data exists for HIV and HCV, where specific KIR genes are supposedly important. For us, we have also showing the functional mechanism behind how 2DS4 is important against the Epstein-Barr viruses. And for immunotherapy, the variants of CD16 and the missing KIR ligand looks to be the most encouraging immunogenetic models to provide information for precision medicine. So with that, I would like to end this presentation here. A lot of acknowledgements, many funding agencies who have funded all this work here in Calgary, my research team, including Dr. Burka, Dr. Storek, Dr. Rad, Poonam and Rehan, Gaurav, Meenal, Amit, so many other people in the lab. Rehan, Arez, and Yassine are the three people who have been consistently working on this NK cell and KIR story from the group. Very strong support from the hematology, hematopathology, and immunology program here. And I'll stop here with this view of Lake Louise, which is a few 100 kilometers away from Calgary, one of the most beautiful spots around Calgary, and happy to take any questions. Thank you.
Thank you very much, Dr. Khan, for this informative presentation. The floor is now open for questions and comments. You can either write your question in the chat section, and if you would like to speak, you can just raise your hand. We'll let you in. So maybe I can start with a question myself. For your BTLD story, you mentioned that TA01 is protective against BTLD. Did you look at this care in the context of matching and mismatching and the degree of mismatch?
Yeah, so the data initially showed the telomeric 1 group is important, but it apparently turns out to be that the KIR 2DS4 is the most important KIR that's conferring the protection. We looked at different matching, mismatching KIR ligand model, and nothing, as I showed for the 3DL1 and HLA-BW4, that it does not seems to be influencing. Presence of 3DL1 leads to protection irrespective of whether the patient has HLA-BW4 present or not. suggesting it's not the KIR ligand, it's more strongly a particular KIR, which is more looks like KIR2DS4, which is interacting with some of the viral antigens and triggering an NK cell response. And if it's not viral antigen, some other ligand on the Epstein-Barr virus infected cells.
All right, any question? I have a question here from Khulud. Says thanks a lot, Dr. Faisal, for the informative talk about NK cell. I think it's a comment more than a question. Thank you, Khulud.
Thank you.
If I may allow Muhammad, just ask a question, just for us to know now. Faisal, do you use now these the NK testing regarding KIR in a clinical practice for routine haploidentical transplant?
So if I understand clearly, you're asking that do we use these routinely for the haploidentical transplants?
Yeah.
Yeah, you know, in Alberta, we are not doing many haploidentical transplants at present. Roughly 90% of transplants continue to be the full house HLA match transplants. We are now starting, working to launch a prospective clinical trial where donors will be scrutinized for the KIR typings and preferably KIR ligand mismatch and a telomeric AO1 group. Donor will be picked among the pool of HLA class 1, among the pool of the HLA matched donors, and hoping to start this study in the spring of next year. And if it continues to be conclusive, then the likelihood is it will become a standard practice to do both HLA and KIR typing for BMT donor selection. But there are many places in states and in Europe where KIR analysis for haploidentical transplant is a routine process, and they like to go for the KIR ligand mismatch donor for that.
Cool.
All right, it seems we have no more questions and or comments from the audience. So with this, we conclude our presentation for today. We would like to thank ARSHI for putting a platform for this activity. We would like to thank Dr. Faisal Khan for his presentation. And we would like to thank all the audience for their participation. And thank you and have a good night.
Thank you very much.